Yeah, once again, welcome to Bio 211D Plant Physiology, Unit 5, Session 6, Factors Affecting Organic Transport. And by the close of this session, you should be able to discuss light and darkness, temperature, effect of phytohormones, effect of minerals, and effect of concentration gradient. Light and darkness. Light has many effects on biological systems, of which photosynthesis is very important. Translocation of food in plants is also influenced by the presence of absence of light. If radioactive carbon-14 sucrose of known quantity is fed to the leaves of two different plants of the same age, and then one plant is kept in dark and the other in light, either for short duration or longer duration, the recovery of radioactive isotopes shows greatest amount of radioactivity in the roots of the plant that is kept in the dark than the plant kept in the light. Though there is a general tendency for the movement of more food material towards roots than shoots, light favors greater movement of organic solids towards shoot apex, and darkness favors the opposite. So the action spectrum of the light that favors translocation has been found to be red and blue, which acts as the action spectrum for photosynthetic reactions. Interestingly, most of the glucose synthesized by leaves during the day is stored as starch, and the same is converted into sucrose at night and transported out of the leaves. This observation can be tested by keeping the plant in the dark for 24 to 48 hours. Afterwards, if leaves are tested for starch with potassium iodide, they do not show any coloration, but the leaves of the plant exposed to light from morning to evening show maximum deep blue color for potassium iodide test than the leaves exposed to light for 1 or 2 hours. The above observations clearly suggest that during night, plants translocate most of their food materials from the leaves to other regions, but during the daytime, they do translocate some food materials, but most of it is stored in the form of starch. Temperature. Translocation is greatly affected by the change in temperature, which means the process is a facilitated process and it needs metabolic energy. If the petiole through which organic materials are translocated is cooled by water jackets, translocation is greatly affected. Similarly, if roots or stem tips are subjected to cool temperatures, the translocation of solutes towards them is severely inhibited. On the contrary, if the temperature is increased, the rate of translocation also increases. However, very high temperature is fatal, but certain cold-resistant plants like sugar beets are capable of adapting to cold conditions by producing antifreeze proteins and maintains the normal flow of food materials even at freezing temperatures. So the effect of phytohormones. Growth promoting hormones like indoacetic acid, gibberellins, and cytokinins are found to accelerate the rate of translocation. As the above said hormones activate cellular metabolic activities, cell division and cell elongation in the apexes and other growing tissues, the cell structures need greater amount of organic solids, energy, and structural components. Hence, these regions act as sink and organic solids move into them with greater facility. So though individual hormones have different but specific effects on plant growth, they accelerate the rate of movement of materials from the source to the sink. For example, IA increases the rate of movement from the leaves to other regions. Cytokinin favors the translocation of sucrose out of the leaves, but preserves nitrogenous compounds within the leaves. On the other hand, if the same hormone is applied to the roots, it favors as well as accelerates the movement of sucrose, amino acids, and other compounds towards roots. So to substantiate the favorable effect of phytohormones on translocation, if an agar block containing IAA, indo-acetic acid, or GA, is placed over the decapitated stem and carbon-14 sucrose is fed to the leaves, the sucrose or the photosynthate found in leaves moves towards the region at which hormones is supplied. So the phytohormones have stronger influence not only on the direction of translocation, they also accelerate the rate of translocation. Effect of minerals. Among all the essential nutrients, boron has been found to facilitate the movement of sucrose. The absence of boron reduces the rate of translocation significantly. Phosphate also facilitates or affects the process of translocation. Effects of concentration gradient. Translocation between the supply end and the receiving end is always governed by the concentration gradient. Steeper the gradient, the greater the rate of translocation, and vice versa. Such a gradient-mediated movement of solutes can be demonstrated by cutting off the leaf, which acts as the supply end. So by removing the leaves, the gradient will be abolished and automatically the transpiration comes to a standstill. Generally, at the supply end, sieve tubes contents show greater positive pressure, and the receiving ends like stem, apex, root, apex, developing flower or fruit show negative pressure. This provides a motive force for the rapid movement of organic solutes from leaves to the other said regions called sinks. So in review, in this session we learned that an increase in temperature increases the rate of translocation of organic solutes and vice versa. We have also learned that the steeper the concentration gradient, the faster the rate of translocation. In addition, the effect of light on translocation suggests that during the night, plants translocate most of their food material from the leaves to other regions, but during daytime, they do translocate some food materials, but most of it is stored in the form.